Thank you everyone for being here. All familiar faces. I'm glad to open formally the spring, that is the spring series of CBAC seminars. It doesn't look like spring yet outside there. It's probably not going to be looking like spring tomorrow either. So we have a real distinguished speaker for our opener of the spring series. It's Dr. Stanley Nattel from Montreal. And because his title is very long, I have to read it because I can't remember it.
Can make it up. Doesn't matter.
Stanley Nattel, MD, Paul David Chair in Cardiovascular Electrophysiology, Professor of Medicine, University of Montreal, Director, Electrophysiology Research Program, Montreal Heart Institute Research Center, Canada. So Stan he told me that was a new revelation that he was born in Israel, and then moved with his parents to Montreal, and received his undergraduate degree and MD both from McGill University in Montreal. He then came to the United States for fellowship training, I think in University of Pennsylvania and then Indiana University.
The other
or the other way. Okay, the order was
glad you're
reversed.
remembering
Indiana
perfect.
University, University of Pennsylvania. And then went back to Montreal where he's been since. And I think everybody here knows Stan works because he touches on every little corner of arrhythmia research and cardiac electrophysiology, both ion channels, remodeling of ion channels, remodeling of cells, remodeling of tissue, atrial fibrillation, atrial arrhythmias with a very strong clinical significance. So today he'll tell us about mechanisms by which exercise training can increase cardiac arrhythmias risk. I'm sure all of you want to hear about it. Can we have too much of a good thing?
Thank you very much. I have to say I'm very pleased to be here, and it's a great honor. I was privileged to see the work of Dr. Rudy's group, a bit of the work of Dr. Rudy's group and of Dr. Efimov this morning, and the quality and breadth are really extremely impressive and humbling. So I'm going to talk today about an area that a couple of my students have been working on for the last few years, and I've entitled it Mechanisms by which Exercise Training Can Increase Cardiac Arrhythmia Risk. Can we have too much of a good thing? So this slide makes one very important point, because whenever I talk about this subject, people say, oh no, should I stop exercising? Is it bad for me? Exercise is a good thing, and this is the risk ratio. This is just one example of many pieces of the literature that say this. This is the risk ratio for death in over 60 individuals per fitness quintile, and to the right are more fit people, to the left are less fit people. These are adjusted hazard ratios. So the lower hazard ratio is where you want to be because your risk of dying is lower. And you can see that there's quite a substantial reduction in risk of dying in over 60 individuals, but I'm sure it's true for under 60s as well, with exercise. So exercise is definitely a good thing. So exercise is good for us, but like most good things, it's not only good. So this is a study from an Italian group which looked at 1,386,600 adolescents and young adults in the Veneto region, which I think is Venice, and 112,790 of them were competitive athletes, which is quite a staggering percentage actually. And so this is the risk of sudden death per 100,000 person years from cardiovascular and non-cardiovascular causes. And you can see particularly the risk of sudden death, of cardiovascular sudden death, is substantially increased in the exercise group. And when you break this down into underlying heart disease, you find that most of the risk is because many of the people who died had some form of underlying heart disease. And therefore, during exercise, they were most likely, if they were going to die, most likely to die at that point during exercise, which acted as a precipitator. So these are people with arrhythmogenic right ventricular cardiomyopathy, coronary arteries disease, congenital coronary anomalies, mitral valve prolapse, myocarditis, conduction system diseases. All the risk ratios are greater than 1, and for some of them they're substantially greater than 1. So for arrhythmic right ventricular cardiomyopathy, there's a 5-fold risk, coronary disease 2.6-fold, congenital coronary anatomy 79-fold. So in somebody with underlying heart disease, a very strenuous training program can be a dangerous thing. So athletics acts as a trigger or a provoking factor on a heart disease background. Now, athletes can also have frequent ventricular ectopy. This is as opposed to sudden death, which is a lethal arrhythmia. These are extraventricular beats, and this is from one individual showing the number of extraventricular beats per hour over the course of a day. The only point is there are lots of them. And again, this tracks heart disease. So this is the group A. So these authors divided their subjects into groups A, B, and C according to the number of ventricular beats. A had the most, B had intermediate, and C had the least. And this population was very enriched by patients with heart disease, indicating again that in patients with underlying heart disease, the risk of ventricular arrhythmia promotion by exercise training is much higher. And to show that these arrhythmias are not simply a coincident observation, but are related to exercise, when they subjected these individuals to periods of at least 3 months of deconditioning, you see a substantial reduction in the number of premature ventricular beats, couplets, which is 2 in a row, or non-sustained ventricular tachyarrhythmias. So the reversibility of ventricular ectopy with deconditioning suggests that the training itself is an important contribution to the mechanism. So what I've talked about until now is ventricular arrhythmias. Now a few words about atrial arrhythmias. This is from a paper which probably by now has been published. I should go back and find out for sure, but it's a review of the relationship between exercise training and arrhythmias, and this specifically deals with atrial fibrillation. And you can see here a list of studies, all of which found an increased risk of atrial fibrillation in highly trained individuals. So this study looked at athletes versus the general population, found about a 4-fold increase. This one had an odds ratio of 2.87, meaning about a 3-fold increase in the risk of atrial fibrillation. This one 8.8-fold. This one found a risk ratio. So these were endurance athletes, marathon runners. This is orienteers, which is a high output form of exercise with a risk ratio of 5-fold. This one in this is the orienteers, and this is the incidence. So the risk ratio was 5.5. Those are the actual percentages. This is in cyclists, 10% incidence of AF versus 0% in control. And this is in just highly active versus sedentary individuals, a 7-fold risk ratio. I don't know how many of you in the audience are physicians? A fair number. So probably you, like I, have seen a fair number of young, otherwise well individuals who are very athletically fit and trained who present with atrial fibrillation, and this can really be a problem. So the risk of AF is clearly, atrial fibrillation will abbreviate AF, is clearly increased in endurance athletes, especially in the 30 to 50-year-old male age range. So that's the clinical background, and I think I've convinced you that A, exercise is very good for you, but B, it can increase the risk of certain forms of arrhythmias in certain individuals, both atrial and ventricular. And now I'm going to talk about an animal model which we developed to study exercise-induced cardiac remodeling, and to see whether in fact there was some increased arrhythmia risk, and if so, how it happened. So this model was initially developed in Barcelona by Begoña Benito, and then she came to my lab and implemented it in my lab, and a major part of the work was done in collaboration with Lluís Mont and Josep Brugada in Barcelona, and then we've continued on, and I'll show you both parts today. So they took male Wistar rats, relatively young because of the increased arrhythmia risk in youngish people, and then divided them into 2 groups. So all observations that I'll show you today, all comparisons are between exercised rats and age-matched sedentary control rats, because obviously with aging, particularly in this age range, they get larger, the hearts get larger, there's developmental changes, and that can affect them. So they did 1 hour daily training 5 days a week for up to 16 weeks. I'll show you some time course data as well. And the exercise training was done on a treadmill in these linear compartments where there's a shock that's given if the rat doesn't run. And this is likely equivalent to about 10 years of high-level endurance training in man. Now, the shock actually, besides being unpleasant and perhaps slightly amusing, is also a problem, because if the rats get a lot of shocks, then it's a stress model and not an exercise model. So we have a 2-week run-in training period during which they learn to run with minimal or no shocks, and then some of them don't learn to run, so we eliminate those, and that's a bit of a bias factor. But the ones who do learn to run get relatively few shocks after that. And this is a little vignette in the study. I've seen some high-quality movies earlier in the day, very sophisticated. This is much less sophisticated. This is what it looks like with our running rats. So, sorry. So, no, we've done that. Okay, so basically then, after 16 weeks of exercise or an equivalent sedentary period, the rats were instrumented under isoflurane anesthesia, and electrode catheters were placed either in the atrium or the ventricle, and arrhythmia induction performed. So this is an example of the induction of ventricular tachycardia by a series of premature beats. And the overall inducibility of sustained ventricular tachycardia, which I believe is defined as 30 seconds, I'm not sure, was substantially higher in the exercise. It wasn't indefinitely sustained, but it was definitely fairly long, was substantially higher in the exercise than the sedentary rats, indicating the development. Now, none of these rats had spontaneous arrhythmias. These are all inducible, but there's a substrate here which can support ventricular tachycardia. In terms of atrial arrhythmias, this is a control rat subjected to burst pacing. These are the stimuli. These are atrial electrograms. This is an electrocardiogram, and you can see at the end of the burst pacing, there is one atrial complex corresponding to a sinus beat and to a ventricular QRS. So these are regular. So this was no arrhythmia induction in a control rat, and this is an exercise rat that after the end of the burst pacing clearly developed atrial fibrillation, which in this case was non-sustained, and this definitely was defined as under 30 seconds. So the incidence of non-sustained atrial fibrillation was 100% in the exercise rats compared to about 40% in the non-exercise, and this was highly significant. So clearly they had a substrate for atrial fibrillation. Now, in terms of longer-lasting arrhythmias, this is an example of induction of sustained AF in an exercised rat. The incidence of sustained AF was 9 out of 14 exercise rats compared to 3 out of 15 controls, and this was statistically significant. And interestingly, the mean duration of AF was very long, considering these are relatively small hearts. The average duration in the exercise rats was 8.7 minutes. So then we thought, well, this is nice, because now we have a model where we can study mechanisms underlying the arrhythmia tendency occurring with exercise. So first we looked at the remodeling of atrial structure and function. And to do this, the rats were subjected to echocardiograms at repeated intervals so we could follow them longitudinally. And so these are the left ventricular dimensions. This is the septal thickness, which is an indicator of left ventricular hypertrophy, as is left ventricular mass. This is diastolic dimension, which is an indicator of dilation, of enlargement of the heart, and this is a normalized intraventricular septum thickness, so it's again an indicator of hypertrophy. So we see that at 16 weeks of exercise, there's eccentric hypertrophy, meaning there's both hypertrophy and dilation of the ventricle. If we look then at left ventricular function in terms of ejection fraction and two indices of diastolic function, again we see a decrease in both systolic and diastolic function at 16 weeks, going along with hypertrophy and dilation. And finally, if we look at the atrial dimensions, we see enlargement of the left atrial diameter in both systole and diastole, with, I guess in this case, about a 25% increase in atrial volume. So 16 weeks of exercise changed both ventricular and atrial function, and not in dramatic ways, but in clearly significant ways.
So
Yes.
those are in negative ways. So this is decompensated, whatever you call it. It's where it's gone into failure, isn't that right?
Well,
There's a
these.
loss of function.
There is a decrease in function, yeah. This is not failure because the left ventricular ejection fraction is still normal for the rat. It's a small change. So they certainly don't go into heart failure, but there does seem to be a negative impact on cardiac function.
Whereas in normal humans, exercise will increase cardiac function until.
Well,
Isn't that correct?
let's say there's an adaptation that occurs with bradycardia and an increased ejection fraction and stroke volume that's largely related to the very slow heart rhythm, and then there's some degree of hypertrophy as well.
Question?
Yep.
Do you have any hemodynamics to go with this in the sense of the control group versus the exercise group?
In terms of filling pressures and things like that?
Mean arterial pressure and stuff like that?
I don't believe we do. These were repeated echo measurements. I'm trying to remember if it's possible that we got measurements at the end of the study with catheters, but I don't have the data with me. But you can't do repeated. Well, you could theoretically have implanted catheters, but we haven't done that. So these are just repeated echoes. Okay, so these are changes now in mRNA expression of various extracellular matrix proteins and important signaling and related molecules. So there's an increase in mRNA expression. Now these are in the four cardiac chambers: right atrium, left ventricle, left ventricle. There's an increase in TGF-beta1, fibronectin, and MMP2, matrix metalloproteinase 2, as well as in collagen precursors, procollagen 1 and 3, in multiple chambers. The left ventricle, interestingly enough, didn't show a statistically significant increase in collagen, but the right ventricle and both atria did. Now here, I put a dashed line around this because it's not statistically significant, but you can see this is probably just a type 2 error because the magnitude of the increase is about the same in RV as in LA and RA. Just it wasn't statistically significant. So these are proteins, TGF-beta1, fibronectin, all the same ones essentially, and a significant increase in TGF-beta1 and MMP2 and collagen 1. There was not a significant increase here in collagen 3. The left atrial increase in collagen 1 protein wasn't statistically significant, but again, you can see that's probably just because of a large standard error bar here. So these are fibrotic changes in the ventricle. These were quantified actually in 3 different ways with Masson trichrome, picroserous red, and hydroxyproline content. Here I'm just showing the picroserous red data, and you can see a statistically significant increase in right ventricular fibrous tissue content, none in the left ventricle with exercise. These are atrial fibrotic changes quantified with picroserous red. This is RA and LA from a rat in the exercise group and the control group. It looks as though there's a little bit more red here. If you look at the overall percentage of collagen, it's significantly increased by about 50% in both the LA and the RA and mean data. This is at 16 weeks. Now, just to put this into perspective, in terms of the role of fibrosis in the atrium, in congestive heart failure, which we've studied extensively, fibrosis appears to be a major factor in promoting atrial fibrillation maintenance. There you see 10 to 20-fold increase in fibrosis. this is only a 50% increase. So while there is an increase in fibrous tissue content, it's not enormous, and perhaps on its own not enough to explain the arrhythmia. And then we looked at electrical and autonomic remodeling. So first, standard electrocardiographic parameters. The only index that was significantly increased was significantly changed was the heart rate, which showed a statistically significant decrease at 16 weeks.
Is this under anesthesia or conscious?
These, I believe, are conscious, but I'm not positive. No, I think they must be under anesthesia because they're ECGs. I think the ECGs are all done under anesthesia. This is in vivo electrophysiological studies, and you can see the resting sinus cycle length was increased, as was the Wenckebach period, suggesting to us possibly an increase in vagal tone. Now, I would say that it may not be optimal. It is, in certain respects, it's not optimal to measure these things under anesthesia. On the other hand, if we want to correlate it with the atrial fibrillation promotion, that was also measured under anesthesia. So at least this gives an index of the conditions under which the arrhythmias occurred. So then, now this actually was done in vivo, consciously, I believe. And this is an assessment of vagal versus sympathetic control of heart rate. So we saw a slowing in heart rate, and the question became, is this due to increased vagal tone? Is it due to sympathetic withdrawal? Is it due to a combination? So what the students working on this did was they, through indwelling catheters, looked at the heart rate under, so this is an actual recording from one experiment. This is the baseline heart rate. This is after administration of atropine to block the vagal system, and this is after administration of propranolol on top of atropine to block the sympathetic system. So atropine tells you the heart rate change under cholinergic block, and then propranolol tells you the heart rate change under both beta blockade and cholinergic block. So the difference between these two is the should be the effect of beta blockade, and therefore it's telling you the extent of beta adrenergic tone. And the heart rate after administration of both beta blockade and cholinergic blockade is the intrinsic rate, the rate that would be present in the absence of autonomic tone. So we did it this way on one day to measure the beta adrenergic tone, and then on another day in the same rat did it the opposite way, gave a beta blocker first and then atropine. So now the difference between the only difference between these two is the presence of atropine, so that gives you an index of the cholinergic component. And so these are the mean data, and it's reassuring that the intrinsic rate measured on two different occasions was fairly similar. There's no statistically so the gray area here indicates the change in heart rate with beta blockade on top of atropine, and it's an index of the sympathetic tone. So that didn't significantly change, although it's a little bit smaller in the exercise rats. But the vagal component showed a substantial and statistically significant increase under exercise conditions. So this suggests to us that the vagal component to autonomic tone is increased. Now, many of you may not be that familiar with atrial fibrillation physiology, but vagal tone is a very strong promoter of atrial fibrillation. In fact, one of the dog models we use to study atrial fibrillation is simply to stimulate the vagus nerves, and then a brief burst of pacing will put the dogs into indefinite atrial fibrillation as long as you continue vagal stimulation. So it's quite possible that this enhanced vagal tone could be contributing to the atrial fibrillation substrate that occurs with exercise. So then we looked at the role of vagal enhancement in AF maintenance, and an obvious experiment to do in studying this is to say, well, if vagal tone is important for maintaining AF in exercise rats, just block vagal tone with atropine and see what that does to the atrial fibrillation. So we looked at its efficacy in terminating AF and preventing AF reinduction, and this is an example of a recording of atrial fibrillation from a rat with sustained AF that had been sustained. I think we took rats only that had AF sustained for more than 15 minutes, so we could be fairly confident that a response was not just spontaneous termination, but due to the atropine. And you can see that after the administration of atropine, there was eventually termination of the AF and restoration of sinus rhythm. So atropine promptly terminated AF in all 8 exercise rats with sustained AF, and to which it was administered, and prevented reinduction in all but one. So clearly vagal tone seems to be playing an important role in atrial fibrillation maintenance. So then the question is, if it's important, why do you have an enhancement of vagal tone? What are the underlying mechanisms that enhance vagal tone? By the way, the notion that vagal tone is enhanced with exercise is not a rocket science discovery here. We know that it's a very old piece of well-accepted knowledge that vagal tone is enhanced with exercise. What's interesting here is I was actually surprised by these results, because I was surprised there was enough of a demonstrable effect in rats that have normally a very high resting heart rate, and that it could actually promote atrial fibrillation under anesthesia. So there are a couple of possible reasons why vagal tone could be enhanced. One is an enhanced vagal traffic coming down to the vagal nerve endings and releasing acetylcholine, and the other is an enhanced cardiac response. So this one would imply an increased rate of acetylcholine release through neural mechanisms, and this an increased response of acetylcholine at the end organ. So we study these separately, and to study the neural side, we looked at baroreflex changes, which is an indication of neural tone. So here, I believe this was done conscious, the students working on this project gave graded doses of either phenylephrine, which is an alpha-adrenergic agonist and increases blood pressure, or nitroprusside, which is a vasodilator that reduces blood pressure. So these were given as boluses, and if we look first just to understand what was done, these are boluses that were given, these are actually the time of peak effect, but these are progressively larger, the effect of progressively larger doses of phenylephrine, and you can see after each bolus you get a gradual increase in the blood pressure, and you can see particularly clearly with the larger doses a corresponding decrease in heart rate. So you're making the blood pressure go up by giving this vasopressor drug, and that causes a reflex decrease in heart rate. And then the opposite, you give nitroprusside, which causes a lowering in blood pressure, and that is associated with a transient increase in heart rate. So, okay. So if we plot these then, if you plot the change in heart rate as a function of the change in blood pressure, and here the results from one sedentary rat are shown by the blue symbols, and one exercise rat are shown by the red symbols, you can see that the tachycardic response is fairly similar between the two rats, but the bradycardic response seems to be greater in the exercise rat than in the sedentary rat. You see the decrease in heart rate is greater for any given level of blood pressure. And I'll show you later some mean data, but this was quite a reproducible finding. The slope of the line relating heart rate change to blood pressure change was steeper in the exercise rats than in the sedentary rats. So then this indicates, therefore, that there's a greater bradycardic vagal response to any given increase in blood pressure with exercise, implying an increase in baroreflex sensitivity, and that would be expected to increase acetylcholine release and produce greater central tone. The other side was looked at by looking at the potassium current produced by carbachol, which is a cholinergic agonist. So these are current voltage relationships. This is actually the carbachol sensitive current. So the experiment was done in the absence of carbachol, and then in the presence of carbachol, the results subtracted to give you the carbachol sensitive current, and you can see that the current is increased with either 10 or 100 nanomolar carbachol. In the it's larger in the exercise rats than in the sedentary rats. So this implies an increased end organ sensitivity to the agonist as well. So both these two pieces of evidence, the increase in potassium current sensitivity to carbachol and the increase in baroreflex sensitivity, implied both neural and organ mechanisms as being involved.
Is that because of you have different amount of potassium currents or
Well, I'll show you, and actually probably not, but I'll show you some data speaking to that right now. So the question is, what enhances the acetylcholine-dependent potassium current? And so we did qPCR of a variety of relevant subunits, and I've illustrated in the bottom diagrammatically the system that we're trying to tease out. So the current, the acetylcholine-dependent potassium current, which is the question you're asking about, is carried by potassium channels that are encoded by two inward rectifier channel subunits, KR3.1 and KR3.4. But this channel is activated when acetylcholine interacts with its receptor, and then this signal is transduced into G proteins. There is an alpha and a beta gamma subunit. The beta gamma subunit dissociates and then goes to the channel and opens it up, operates it to increase the current. So first we measured the expression of IKCH channels with PCR and with quantitative PCR, and you can see that if anything there was a reduced current with exercise. So the open symbols are sedentary, the closed symbols are control, and there's a reduced expression of the potassium channel. So it doesn't seem to be due to an increased amount of potassium channels, at least based on the mRNA measurements. We then looked at a variety of adrenergic and cholinergic receptors. So the M2 muscarinic receptor, which is the one that responds to acetylcholine to produce IKCH, there's no change in its expression, and similarly there's no change in the expressions of alpha-1, beta-1, or beta-2 adrenergic receptors. So that's unchanged. We then looked at the G proteins, and the principal cholinergic alpha subunit, G-alpha I, is not changed. There's no significant change either in G-alpha S, which is the principal sympathetic G protein, nor in the beta or one of the gamma subunits. There's a small increase in G-alpha Q, which might be of significance, but that was the only change we saw. So the principal muscarinic coupled G proteins were unchanged. So then we looked at another set of proteins called regulatory G protein subunits, and this is an interesting set of proteins that have GTPase activity and regulate the function of the G proteins that respond to cholinergic stimulation, as well as a variety of other G proteins. And interestingly, almost all of the different forms of RGS subunits were downregulated. RGS 3, 4, 10, 12, and 16. I think one was upregulated that's not on here. Now, the function of RGS proteins has been studied, and at least some of them seem to mediate vagal desensitization. So when you stimulate IKCH with carbachol, you see a progressive decrease in the amount of current despite the continued presence of the mediator. But if you knock out RGS4, as was done by Scott Hexamer's group in this study, you see a longer lasting response and a greater amplitude, and a substantial and statistically significant decrease in desensitization. So right now we don't know for sure why the response to acetylcholine, the potassium current response in acetylcholine, is increased in exercise rats, but RGS protein downregulation is an attractive explanation. Certainly quite striking. Okay, so I'm going to finish now by talking about the reversibility of remodeling. How reversible are these changes, and how long does it take for them to reverse? So these are measurements, these are actually reversibility of changes. This should be ECM-regulating and ECM protein mRNAs. I believe these are all mRNA levels. So these are different cardiac chambers, right atrium, right ventricle, and left ventricle. and the bars indicate statistical significance of changes. So you see that with exercise, there's an increase in TGF-beta1, fibronectin, MMP2, TIMP1, procollagen 1 and 2 mRNA. That's shown by the red. And then within 2 weeks of discontinuation of exercise, all of these mRNAs decreased significantly in their expression level and went back close to or at control. So basically, I should have explained, the way this experiment was done was to take sedentary rats actually matched with each of the subgroups and compare them to 16-week exercise rats or rats exercised for 16 weeks and then allowed to decondition for 2, 4, or 8 weeks. So you see that the mRNA changes recover very rapidly after discontinuation of exercise. Now, what about hypertrophy and right ventricular fibrosis? Obviously, the mRNA is a proximal signal to whatever is going on distally, and clearly there's not a one-to-one relationship, even with protein, let alone with overall functional changes. So this is heart rate over body weight. It's the simplest measure of cardiac hypertrophy, and this is right ventricular fibrosis, and this is right ventricular hydroxyproline content, which is an indicator of fibrosis. And so with exercise, you see an increase in heart weight to body weight ratio reflecting cardiac hypertrophy, and right ventricular fibrosis reflected in each of the 2 measures that we obtained. And you can see that there is some reversal, but it's much slower than what I showed you for mRNA. The daggers indicate a statistically significant change from peak exercise. These are the exercise group, or these are the sedentary groups going with each of the exercise or post-exercise deconditioning groups. So you can see that and the asterisks represent difference from the matched control group. So you can see that for left ventricular hypertrophy, for cardiac hypertrophy, it took 4 weeks to see statistically significant decrease from the peak change at end of exercise, and even after 8 weeks, there was still an increase relative to control. For right ventricular fibrosis, there appeared to be full recovery at 8 weeks, and hydroxyproline content, again, statistically significant recovery, but it still took 8 weeks to see a statistically significant reversal relative to peak exercise. So the actual cardiac structural changes of hypertrophy and fibrosis reverse much more slowly than the mRNA changes following exercise cessation. Now, in terms of the atrial arrhythmias, these data that I'm going to show you are actually, they're not final data yet, but they're the data that we have as of last week, because these studies are still ongoing. But they give you some interesting insights, or some interesting potential insights into the relationship between AF inducibility and potential underlying causes. So, for example, we see that AF inducibility, percent sustained AF, or any AF, which is sustained and non-sustained, as I showed you before, is substantially increased at 16 weeks. By 4 weeks of discontinuation of exercise, there's the AF supporting substrate is largely reversed. So recovery from the AF substrate with exercise is very fast. If we look at atrial size as an index of structural remodeling, left atrial diameter was still not fully recovered after 8 weeks of discontinuation of exercise, and right atrial diameter, although it fully recovered at 8 weeks, was still significantly increased in 4 weeks. So we see that atrial enlargement reverses slowly, and reverses much more slowly than does atrial AF inducibility. So atrial enlargement, at least in itself, is not enough to explain AF inducibility. We can't exclude a contribution, but it appears not to be the whole story. Autonomic tone, on the other hand, we see very rapid reversal of the increase in parasympathetic tone, which this is based on the atropine plus propranolol experiments. We see that by 4 weeks of exercise, it's completely back to baseline.
Why is the sedentary changing so much? Is that age?
Well, it could be. It's a good question. Could be age. These are different groups of animals, but Yeah, I don't have the statistics on whether this is a statistically significant change. But it could be aging. It could be that aging is changing the autonomic tone. And this is the baroreflex sensitivity data. We actually haven't done, and I don't think will do, at least for the moment, the ionic current measurements. But you see that the bradycardic reflex, which was substantial, the slope of the bradycardic reflex, which was significantly increased at 16 weeks, by 4 weeks of stopping exercise is no longer different between the two groups. So both the baroreflex response and the vagal response seem to reverse very rapidly, corresponding to the change in atrial fibrillation inducibility, and agreeing with our data on atropine administration that vagal enhancement is a very important component of the atrial profibrillatory substrate associated with exercise. So, in conclusion, exercise induces significant atrial and ventricular remodeling in terms of chamber size and function. Remodeling is associated with increased inducibility of prolonged ventricular tachyarrhythmias and atrial fibrillation. Structural changes likely contribute to arrhythmogenesis, although we can't say for certain that they're essential. Autonomic changes, particularly vagal enhancement, contribute importantly to AF promotion. Structural remodeling appears reversible but relatively slowly. And finally, RGS protein downregulation may be a key molecular contributor to enhanced vagal tone, at least at the end organ level. Thank you very much.
Have you looked at any stress kinases, any pathways involved in
Which proteins?
P38 isoforce protein?
Oh, no, we haven't.
We found recently, could be relevant, I don't know, that in a mouse when you knock out PYK2, citrulline-rich tyrosine kinase, which is upstream of P38 and participates in mechanical response, mechanical stress. If you knock it out, you actually gain significant vagal response in the ventricles, and it's highly pro-arrhythmic. So it would be interesting to explore, at least at P38 level, maybe upstream in the mechanotransduction stress produced by overload of exercise. So it would be interesting next step.
Yeah.
So one question is that I'm not familiar with the measurement. So we know that heart exercise is good for heart in general.
Right.
So what do people measure in the structural function change in the heart when they actually got improved by exercise? And then did you measure those parameters to see your exercise?
I don't know when to say that exercise is good for the heart, which I definitely agree with. I'm not sure that's mediated by an increase in function per se. It's more mediated by a resistance to injury and a resistance to ischemic insults. The typical changes that happen with exercise are a slowing in heart rate and an increase in stroke volume and an increase in ejection fraction. And the main mechanism, if you slow heart rate, stroke volume and ejection fraction have to go up or cardiac output will go down. So a big part of the increase in that function is simply the Starling relationship, because if you slow heart rate, the diastolic filling of the heart increases with each beat, and so the force of cardiac contraction is enhanced. I don't know, Gene knows a lot about this stuff.
Yeah. I have a question actually about the fibrosis.
Yeah.
So, is there a correlate of that in other models of exercise, and is there any data from humans to suggest that are you suggesting that the fibrosis is what underlies the arrhythmias? It's structural, you think?
The fibrosis happens. It could be a contributor to the arrhythmias, particularly I think at the right ventricular level. And is there a clinical correlate? There are a couple of clinical correlates. The most direct one is that Matthias Friedrich in Calgary has done gadolinium enhancement studies in highly trained athletes, and he does find any evidence of an increase in fibrous tissue content. I think he's looking at the left ventricle, but I'm not sure if it's published yet. I know it was presented, and so that's a pretty direct clinical correlate. The other thing is we know that arrhythmogenic right ventricular cardiomyopathy is particularly prone to interact with exercise, and it's even been proposed that exercise in some individuals produces a kind of acquired ARVD pathophysiology.
Yeah, I think the question is whether if you have the substrate and then you have.
Right.
Yeah, versus
Versus if you have nothing. So at least
Exactly.
Matthias Friedrich's data would suggest that there's some degree of fibrosis that's produced. Now remember, these rats, it's very hard to estimate an equivalent between rat and human exercise. The lifespan is so different, and if you correct for the lifespan of a rat versus a human, the duration of exercise that we expose the rats to is about 10 years. And it's about 80 to 90%, it's 85 to 90% of peak oxygen consumption. So it's very high level exercise training for 10 years. Now, but I don't think you can say that they're directly equivalent. So, I mean, I think...
you know that in the physiological hypertrophy model we look at, it's a mouse, not a rat.
Right.
But there's no evidence, Casey can correct me if I'm wrong, but there's no evidence of fibrosis. So that's
based on...
based on histology.
Uh-huh.
Yeah.
So, it's the same question of whether it's getting into pathologic over-exercise.
Right.
And I'm wondering, you didn't show us the data on the function, cardiac function after decompensation. So do they recover? That slight loss of function that
Right.
you had.
That's a good question. We don't have the data right now. I believe it's being gathered, but we don't have the data.
Did you see?
Well, we have the data on the atrium. I think the ventricular data haven't been analyzed. So with the atrium, it seems to recover. If the atrial dimension recovers, I'm guessing the ventricular function has to recover too, because the reason the atria dilate is probably because of diastolic dysfunction in the ventricles. Yeah.
So is the tissue level of ANF or BNP increasing the exercise training?
I don't think it is.
It isn't.
I don't have the data here. Actually, I have the data on my computer, but I'm pretty sure it doesn't.
Because
Because we've done TLDA on these rats, so we have a lot of data. And if you want, I can look for it.
so can I have another question?
Sure.
So I saw the animals with exercise training have LV dysfunction, diastolic dysfunction,
Right.
and but they don't have a fibrotic change in the chamber of LV. So is there any explanation to that? That they don't have histological change, but they do have functional diastolic dysfunction.
That's a good question. I can't answer it. Yeah.
It's nice talk. I have two questions. First is, as I know, acetylcholine sensitive K channel is usually expressed in atrial, right? So, but you also find there's some ventricular arrhythmia happen
Yes.
in this rat. So
Yeah,
how
the
to explain-
ventricular arrhythmia we don't think has anything to do with the vagal tone.
Okay.
And the QRS was actually slightly but significantly longer in the exercise rats. It wasn't in the data I showed you here, but I believe that in the final data it was. So there may be either slight conduction slowing or, well, it could also be due to cardiac enlargement, maybe. I don't know. So there might be some slight conduction slowing in relationship to the fibrosis which is involved. But the ventricular arrhythmia mechanisms we really don't understand well.
Okay, then second question is, do you ever observe some other kind of current in this kind of
Yeah.
nerves? Okay, is it some abnormal?
Okay, remember we've only studied the atrial ion currents. And there are changes I'm trying to be sure. I'm pretty sure there's a change in a number of the currents and they're balanced. I have the data in the backup. Here's ITO and IK1, so they're both decreased. And I know it's different from your results. Well, these are normalized data. Sorry. Yeah, exercise is decreased, and IK1, yeah, it looks like it's increased. I'm okay. You're right, and I'm not sure if that's real or not. As I say, these are data which I got last week and didn't have a chance to go over. I think it was decreased. We have the subunits. I think they correlated with this. It's possible that the symbols were just reversed here. I know that at one point they had reversed, my students had reversed the symbols. Here's another, yeah. Here are the subunits measured on TLDA. So KR 3.1, 3.4 I already showed you. KB 4.2 and 4.3 are down, as is KCHIP 2. These are all normalized. So. And the KR is not changed. So I believe that may be an error in representation. I think the calcium was measured as well. I don't have it here. But I don't think that the intrinsic, the ERP was not significantly changed, so I don't think the intrinsic action potential duration was altered. That's why my vague recollection is that the potassium currents were downregulated, as was the calcium current, but I'm not positive.
My question was related to the predominance in male populations that you see. I mean, do you think that's related to how much they exert themselves relative to females, or if it's due to some kind of, hormone changes?
Yeah, that's a very good question, actually, and I've never thought about it, but it is really an extremely good question. I believe that females are supposed to be somewhat resistant to atrial fibrillation, although there's different results in different studies. And so whether the fact that there seems to be a particular correlation, particular AF diathesis in males based on the data that I showed is related to the intensity of the exercise, or it's related to a true sex difference in risk of atrial fibrillation, either with exercise specifically or in general, I'm not sure. I can tell you that in my clinical experience, all the patients I've seen with exercise training-related atrial fibrillation have been males. None of them have been females. But that's not a very reliable sample because I don't know how many females, particularly in that age range, exercise to the same level as males.
Well, the men just complain more.
Well, I have to say, most of these patients are not particularly complainers. They had real problems. Yeah.
So I was going to ask you about the basic physiology of the right heart versus the left heart. The right heart being classically in physiology taught as being primarily a volume pump.
Right.
It does very little pressure work because the incoming pressure is low, the outgoing pressure is low. And so your model of hypertrophy here is presumably a volume-related model of hypertrophy.
Yeah,
well, that's why I asked my hemodynamics question.
Right.
Which is, to the extent that pressure is contributing to the changes, if the hemodynamics were available before, during, and after, you could tease out the extent to which the volume response versus the pressure response can differentiate between what you see. The fibrosis in the atrium is interesting because one of the protective mechanisms that the four-chambered heart has for increased volume load is fibrosis, to prevent distension. And it's the fact that it's reversible on the usual time scale, whatever the rat time scale, shows that it can come and go. And so the volume, presumably the decrease in atrial stress due to decreased volume.
I'm particularly interested to know what actually happens to fibrosis. We haven't measured fibrosis per se. The changes in mRNA just tell you that there's less collagen gene activation, but that doesn't tell you that the fibrosis is reversing. And the change in volumes also doesn't tell you either, because I know that we've done studies in the heart failure dogs where we induce heart failure and then allow them to recover, and the atrial volumes come back almost to normal. It's perhaps a slight persistent increase, whereas the fibrosis doesn't recover at all.
So in this model, do you track total four-chamber volume as an index of hypertrophy?
Well, each chamber is analyzed on echo. Actually, I don't think they see the right ventricle very well in terms of volume, but they certainly see both atria and the left ventricles. So we'll have more information on that when the deconditioning data are analyzed for the ventricles. I showed you the data for the atria. We are getting formal and fixed tissues, but as I said, these studies are still ongoing. I presented you data that's kind of in the middle, and that's why it's kind of flying, and unfortunately there may have been a labeling error for the IK1. I'm curious to go back and check now. But we're only going to analyze the fibrosis data when we have all the samples, because the fibrosis measurements are potentially subjective and subject to error, and I don't want to have 2 batches done at different times. I want them to get all the sectioning done at the same time and then do all the analysis at the same time. Yeah.
So actually, my question related to modeling as well. So, what if you find something interesting here? Can you use transgenic approach rather?
We don't have access to transgenic rats. I think they become theoretically doable, but no.
So what can you comment on other exercise-induced models? Is that possible? Maybe some animal model
Yeah,
that
it could be possible in the mouse model. Obviously.
mouse model and
We've got a mouse exercise model, but I don't know if they have an AF susceptibility. Frankly, when this student started to do this work, I was not optimistic for 2 reasons. First of all, the rat seems to have a relatively small vagal tone, and it was my preconceived notion that vagal tone has an important role in AF. And the second thing was that we had to do the electrophysiology studies under anesthesia, and I felt the anesthesia would most likely interfere so much that you wouldn't be able to unmask a change. I was actually surprised to see the results.
So maybe correct me if I'm wrong. So what I saw was that for the protein and the gene expression, left ventricle is very different from other. So one thing is that what's the significance of that? And another question is.
I'm sorry. What do you mean?
One question is what's the significance of that difference?
You mean why is the left?
Why it is, and is it doing anything to the the arrhythmia that you see? Another thing is that so if you see different muscle, different chamber have those differences. Do you think that in the same chamber you can see also differences in different parts of the
regional
muscle?
differences?
Yeah,
okay.
regional differences. So that's actually how would that contribute?
So the second question is almost a religious question because I don't have any data on which to answer. So I'll just say, there could be regional differences, but beyond that, I have no data, so I don't know. As far as the issue of why the difference between the left ventricle and the other chambers, our thinking is that most likely it relates to chronic exercise is believed to be mostly a volume load, a volume stress, and the atria and the right ventricle are much more susceptible to volume loads because they're thinner walled than the left ventricle. So that would be my supposition as to why you see bigger changes in the other ones than in the left ventricle. But
Do you see that at the cellular level? Difference in cellular hypertrophy between the right and the left ventricle in this model?
we haven't looked at that. Yeah.
I have a question about reversibility. How is the reversibility of functional change?
Yeah, that was asked.
In terms of diastolic and systolic.
Right. I don't know yet. Those echoes haven't been analyzed. So the echoes have been analyzed for the atrial component, but not for the ventricular one, which would be quite interesting to do, I agree.
Any other questions? Here's another.
I have a question regarding your title. So exercise is good or bad for us? So do you
I think you would have to work very, very hard to do harm to yourself with exercise. How's that? That's not completely true. You can definitely damage your musculoskeletal system substantially if you're not careful. But as far as the cardiac system is concerned, I think the risk of not exercising is vastly greater than the risk of exercising. But having said that, certainly there are individuals who have major problems with atrial fibrillation with high-level training, and so there you have you have to decide: do you have them decrease the level of training, stop training? Clinically, often they're given antiarrhythmic drugs, subjected to AF ablations and things like that. I have one patient who came to me with AF during a period where he had started to do extremely intense physical training, a lot of weight training and things like that. His heart rate was extremely slow, and I suggested he slack off a little bit, not stop his training, but maybe decrease the level. and I put him on an antiarrhythmic drug, and his atrial fibrillation got better on the drug, and then later I was able to stop the drug, and he stopped having recurrences. So for people who, and not, you know, people get addicted to exercise, and I think it's both the physical well-being plus the endorphin release associated with it. So not everyone is going to be willing to decrease their level of exercise, even if it's causing them some problems. Yes, Jörn?
So early repolarization is more common in athletes than in the normal population, and you know it's always been viewed as benign, except now with Hasegaard studies and so on, it's not so benign.
Right.
And it's hard to separate the normal population early repolarization syndrome from the athletes. Do you see any? Have you looked into any possibility?
I had my students look at the rat ECGs, and they didn't find any evidence of early repolarization. But I have to tell you, there's a very interesting animal model which is very relevant to all of this clinical issue, which is the racehorse. And apparently, atrial fibrillation is a very big problem in racehorses because when they get it, they can't race and they have no commercial value. So there is an intrinsic model, and they, of course, have extremely high vagal tone. They also, interestingly, get ventricular arrhythmias, and I think they get in association with early repolarization. I don't think that, to my knowledge, they don't have sudden death, but I'm not sure. But they definitely, I happen to be a thesis examiner on a DVM thesis at a veterinary school, someone who developed an atrial defibrillation technique for horses. Major commercial value, although intellectually it may not be earth-shattering. And so I was in contact with them afterwards about a lot of these things, which I found very interesting in their population. And her supervisor sent me a paper that he wrote about ventricular arrhythmias in racehorses, which apparently can be quite significant.
So these clinical studies you showed us, it seemed like a lot of the exercise subjects were marathoners and these really elite athletes. So it's kind of a simple question, but are there any behaviors that these athletes might have that would actually predispose them? So let's say, would they abstain from certain cardioprotective behaviors?
Well, clearly as a group they're a selected group. And my guess is, if anything, they're more likely to engage in cardioprotective behaviors than abstain from them. So I can't think of any. I'm sure there are alternative hypotheses to explain, mechanistic hypotheses to explain some of these things, but I can't offhand think of any obvious ones that might. So I don't know. I mean, certain kinds of athletes might smoke more cigarettes or drink more coffee, but many don't. And a lot of these kinds of studies have been done in orienteers and cyclists and people who are not the population you think of as engaging in behaviors that might be damaging to the heart. So I don't think that that's the explanation. I think clinically, certainly, the atrial arrhythmia story is much cleaner and clearer than the ventricular arrhythmia story. As far as ventricular arrhythmias are concerned, it's clear that for certain kinds of cardiac pathology, people who engage in high-level exercise can worsen that pathology, and the classical example is ARVD. But ventricular arrhythmias due to exercise in someone with an otherwise completely normal heart, I think that that's less of a concern. Other than this fascinating and totally unclear picture of early repolarization, where I don't think anybody knows at this stage whether someone who has severe early repolarization from intense athletic endeavor is likely to get a ventricular tachyarrhythmia. There have been studies which have followed athletes long term, and I think their sudden death rate is lower, if anything, than the population as a whole.
I was wondering if there could also be an inflammatory component. Rich Schuessler here is looking at inflammation in AF, and I guess athletes also incur a lot of injuries, even if they're low level. I'm wondering whether or not that might play a role, particularly since you saw TGF-beta and other.
That's possible, although interestingly, in heart failure models, exercise seems to reduce the markers of inflammation in the heart. So I'm not sure which way it really would go. Yep.
So this exercise model was produced in the control rat, right? So can you comment on like what if the heart already has some substrate like in functions, and what exercise can?
Yeah. So the data I'm aware of, there's been work, I believe in both rat and mouse models, that show that exercise appears to antagonize adverse remodeling post-myocardial infarction. And also, I believe that exercise reduces the sudden death rate upon acute myocardial infarction. And I suspect that's kind of a mimicking of cardiac preconditioning, because exercise engages vagal tone and GI proteins engage a lot of the same signaling systems as a variety of forms of cardiac preconditioning. So I think that probably the exercised heart is at least biochemically preconditioned against ischemic insults. Yeah.
And following the same question, what if you put a pacemaker in this rat and pace it 18 for 2 weeks or 3 weeks? Will you get a similar response? Fast rate?
You mean if you prevented the bradycardia?
No, if you pace at a rapid rate.
Oh, pace the atria at a rapid rate, would there be a difference? Again, that's a philosophical question. I don't know, not having done it. If you can get me rat pacemakers, I'm sure if anybody can, you can.
Yeah, we just made a mouse pacemaker.
Maybe too small for a rat.
Well, it's probably more interesting to do in the mouse than all your transgenic.
You're probably right that if mouse area too small to sustain atrial fibrillation.
Well, that's not true. I mean, we know that there are genetic models of atrial fibrillation in the mouse. So one of the things that people forget when they talk about the size of the heart in relationship to ability to fibrillate is that it's related to the size of the heart, but it's also related to the action potential duration. And because the action potential is so extremely short in these kinds of animals, small rodents, it's not at all impossible for them to fibrillate.
Okay, we can see that medicine is getting very specialized. In addition to that, before we open, so there will be a reception buffet out there. Oh, before we talk about the buffet, have you considered giving the running rats cheese instead of giving shocks to those that don't?
It's a question about how much a rat will run for cheese. Maybe if you put a female rat in the other end, it'll run. Then only male rats, presumably.
Stan, I didn't want to go there. Montreal.
Yeah, well, you know, the French.
All right, thank you.
Yeah, that's right.
Oh, really?
Yeah, I remember I was there, like, Aaron, and he's like,